It's been a long road, but I think we are making some progress. So I'm pleased to be here today and be able to share that with you, to talk about where we've come from, but more importantly, where we're going, because we still have a long way to go to make sure that HCU is a very manageable disease, if not non-existent. And so you've all seen the pathway. How many of you feel like you're prepared to give this pathway presentation yourself? I'm only showing it because I'm going to set up the potential treatments in the context of this pathway so you can see where they work. So I assure you I am not walking you through the pathway. So when we look at what approaches are being studied for HCU, and I should say as background, those of you that have been at these meetings before will know that we started out in 2016 with a research map like Brittany just presented for the remethylation disorders. And a lot of this research wasn't underway. And so I'm thrilled to have 5 different areas where products are in some stage of development. And so this describes the mechanism or the type of program underway. And so if you think about the pathway, we all know for CBS deficiency, the main bottleneck or blockage is the lack of an effective CBS enzyme. So first and foremost, can we replace that enzyme so that patients can break down homocysteine? And there are 2 approaches. I think last time we met, there was only one that was being studied, but it's great to see that there are now 2. And the first you're going to hear a lot more about from the next speaker, but that's replacing the enzyme directly. And the second is presenting an alternative enzyme in patients that's using a bioengineered enzyme that also breaks down homocysteine as well as homocysteine, which, if I remember correctly, is the dimer, the 2 combined in that little graphic that I think Kim showed us. So you won't be hearing about that today, but hopefully at the next meeting we're going to have more progress on that approach. Second category, definitely new in the last year or 2, and that is oral therapies that can be taken and they work in the gut to prevent methionine from being absorbed. So imagine, I don't know if you take this maybe before a meal, but let's say you or your child takes this before a meal and then the protein that they ingest, the methionine is broken down so it never gets absorbed. That sounds like a pretty neat approach to me. And I'll tell you, there's 2 different approaches within that category. The next you've heard about in the past or you've heard about today, and that would be gene therapy. Instead of delivering the enzyme directly, can you deliver the DNA that codes for that defective enzyme and enable the liver cells to produce the enzymes, like having your own little manufacturing factory within your liver? The next category is chaperone therapy, and that would be administering a compound that can combine with the defective enzyme. And I know, we don't go into great detail on that usually, but to say that someone doesn't have CBS enzyme, they really don't have a functional CBS enzyme. They may still make the protein that forms, an enzyme is a protein that folds into a 3-dimensional structure. But what happens if you have a genetic defect is it doesn't fold properly or it folds and immediately unfolds. And so a chaperone would be able to combine, think of it like a lock and key, and stabilize that defective enzyme. And the last are other approaches to modify the metabolic pathway to help lower homocysteine, and I'll explain about that on a subsequent slide. So again, here's the pathway, and this just shows where each of those potential therapies would exert their effect. If you go to the bottom right, can you directly replace CBS with enzyme replacement therapy, with gene therapy, or with chaperone therapy, you don't replace CBS, but you make CBS function? Then you could go to the lower left and have an alternative enzyme that replaces that defective CBS and does the same thing metabolizes homocysteine to cystathionine. You could go to the upper left, where I explain the GI therapies and prevent methionine from being absorbed, so you won't have a lot of methionine to convert into homocysteine. Or you can not modify the metabolic pathway either, between the methionine homocysteine part or down directly at homocysteine, and I'll explain that more in a moment. So now I'm going to go through each of these 5 categories and give you an update on the status of the program. And the first is enzyme replacement therapy, and the name of that compound is pegtibatinase. We'll see if you hear more correct pronunciation in the next talk. But that's taking the CBS enzyme, that's made by a bacteria, and adding this pegylated compound. I would like to note this is being developed by Travere, but I would like to give credit for the discovery to Jan Kraus, who was a phenomenal researcher, very committed to homocystinuria. We would not have this program today were it not for Jan and his collaborator, Tomas Machen, at the University of Colorado. And I met Jan back in 2009, 2010, and I was very pleased when I told him I was setting up a research fund in honor of my sisters, and he said, I have a program that could use some funding. And so I was pleased to be able to give him a grant and to fund the mouse study that proved that it worked, that led then to a different biotech licensing it in, and Travere bought those rights and is now bringing the program forward to development. So phase 1, this is called 1-2A. You might have heard Kim Chapman talk about 1 separate from 2. Sometimes you can combine them. Phase 1 usually looks for safety, and 2A looks for dose response to see, efficacy dose relationship. And they were able to do both in the first phase by starting with a lower dose and increasing that. And so that was started in early 2019. COVID slowed it down a little bit, but the company stayed committed, and they released positive data the end of next year that you'll be seeing, the end of last year, which you'll be seeing in the next presentation. And there's an additional dose cohort they've added on to see if they could get even higher efficacy. The estimated timeline, this is purely coming from me. This is not the company's. This makes the companies very nervous. They only like to tell you exactly where they are right now and maybe, data expected end of the year, et cetera, because they don't want to have investors angry at them if they don't meet their timelines. But I know from 26 years in the industry what the usual timelines are, and I've added on a little bit of cushion, and we could see a product getting to market if all goes well in 2023. So that's not too far away. It might be, I'm sorry, I didn't mean getting to market. Getting into phase 3, and that should happen because they're in discussions with the FDA now, but getting to market in 2026. So 4 years from now. So just keep your kids on that low-protein diet, betaine, everything else they're on, and maybe life will get easier. Then we have the alternative bioengineered enzyme. Doesn't yet have a name other than AGLE 177, coming from aglia. That's where the AGLE comes from. They say it's a homocysteinase enzyme. It breaks down homocysteine. And it's a mutated form of a different enzyme that, they do all their fancy, bioengineering with and come out with this product. Phase 1 and 2 is also underway. They started later, but they opened sites in UK and Australia first, and they're now opening sites in the US. They did disclose positive results in the initial dose cohort, but they're going higher. And they have announced that they expect clinical data also before the end of 2022. And so the timing could be, similar if both companies get the results of their first phase of data at the end of the year. Both could be in phase 3 in 2023, and both could get to market in 2026. And if it's one thing I learned 26 years in the industry, competition is good for patients. You should all be happy because they not only want to get a product to market, but they want to beat their competitor. So that's always good for patients. And who knows, maybe there could be some price competition, but I know they can't talk about that. So the next category, I'm thrilled on that progress, by the way, that we could potentially have 2 products within the next 5 years available for patients. The next category, the oral GI therapies. The first one that I'll talk about is from Codexis, and both of these companies will have posters. So make sure during the reception you listen to their poster presentation, because both have data and animal models that give you good reason to believe it could work in humans. And so the very first one is an enzyme that is somehow encapsulated and taken orally, and it degrades methionine. They are doing the studies right now that you need, such as toxicology studies, to get your approval from the FDA of your investigational new drug application, which means you can go into humans. Both, as I said, had positive results in a mouse as well as non-human primate studies. So based upon where they are now, phase 1 could begin in 2024, which could then take you out 4 or 5 years for a launch. The second one is actually a probiotic, and you may be familiar with yogurts that are considered probiotics. And they engineer this so that the bacteria will consume methionine, and this is Synlogic's product. They are at IND filing, and they've announced that they expect to begin phase 1 in 2022. And they're doing that in healthy volunteers. My slide originally said normal volunteers, and I got slapped by Danae. She said, "Are you saying that HCU patients aren't normal?" And I said, all right, Danae, I get it. Okay, so I will never, ever in the future say normal volunteers. Healthy, non-CBS HCU patients, phase 1 will be done. And you can test. I mean, the interesting thing here is normally if you do healthy volunteers, you're not able to see if there's an effect. You do it to see safety. But here you can't see if there's an effect, because what you're looking at is when you give them protein, are you preventing the absorption of it? So this phase 1 for either of these programs could tell you whether it works as well as whether it's safe. So maybe that'll speed things up. Hard to tell. And I forget which company. One of these companies is doing the same program for PKU, and it's a year ahead. I kind of think that's Codexis, but you can ask them at the poster. So that's great because they're learning from PKU, so that also could make the timelines quicker for HCU. Next is gene therapy, and this program is taking the DNA that codes for the CBS enzyme, attaching it or putting it in a vector that's called an adeno-associated vector, which is a modified cold virus that has no biologic effect. And this has been tested in mice, and this is a study done jointly by Warren Kruger, who has a mouse model at Fox Chase, and Ron Crystal, who has done a lot of gene therapy development programs at Weill Cornell. And I gave a grant directly from the Hempling Foundation years ago for this. I now mostly give the money to HCU Network America that goes then through the normal grants process, but this one had been done prior to that happening. And so the results were very compelling in the mouse model. They have been presented at a scientific meeting, and they are finalizing a funding source for collaboration with a partner who I don't think plans to announce who they are at this stage, but if they're here, thank you. And we look forward to hearing more about this, and then they could, that will then fund getting into the phase 1 trial, and earliest based upon not knowing exactly what needs to be done and when the funding will begin is maybe 2025. Chaperone therapies, I explained that kind of lock and key approach. That work, we actually just gave a grant out through HCU Network America to Thomas McCorvey in the UK. And he is using a very high-tech approach called fragment-based X-ray crystallography. If anyone in the room knows what that means, good for you. But it will screen for molecules. It's all about structure. I mean, it is kind of intuitive that if you want something to be able to combine with something else, they have to have the right structures. And so they will screen for the right structure and then see if it restores enzyme function. If it does, then they would need to go on and test this in cell lines and animal models. So we can't even begin to estimate how long that would take, but it's good to see that's being done. It's also being done for cobalamin C with another grant you'll hear about in a moment. Then there's two programs that fall under the metabolic pathway modification. One is Formate, done by Ken McGlynn, who I know some of you are familiar with at University of Colorado. And Ken was awarded a grant through HCU Network America and HCU Network Australia, and separately gets funding from the Will Hummel Foundation grants. So thank you, Hummel family, for, where's Will? Where's Will? Oh, Will's at the grocery store. When Will gets back from that, if anyone wants to thank him for, I think it was his grandfather who provided that funding, but it enabled Ken to be able to do everything he needed to do to show that low-dose Formate could safely lower homocysteine in his mouse model, and very exciting results that when it was combined with betaine, it achieved near normal levels in the mice. And so Ken is very excited to take this program forward. He needs to determine exactly how do you deliver that Formate. Does the combined therapy improve cognition, bone density, prevent clotting from occurring? He did receive some NIH funding, and he also is very close to signing a deal with someone who's going to fund this and help it get further along. And if they do announce who is funding it, we will make you aware of that through the newsletter. And then the last program was a program that was funded through HCU Network America combined with HCU Network Australia. And that was looking at a way to increase the bioavailability of free homocysteine that's not in a bond, to form a dimer or not binding with a protein, because the enzyme only works on free homocysteine. And so Tomas had this, proposal that he thought if he were able to increase free homocysteine and then add a form of ERT, it wasn't the exact one that Travere is working on, but close enough that he thinks it's representative, does that lower HCY? And he showed in mice that it did. And so he doesn't have a timeline just yet. I think he is looking for funding and still continuing to do some work. But if it were me, I would wait to see how much lowering do you get from Travere's program. And if it's enough, you don't need a second compound. It only complicates things. So this may come into play if either overall the program doesn't get high enough efficacy, or perhaps there's some subset of patients where you don't get enough efficacy. So this could be a supplemental therapy down the road, but that's to be determined. So back to the metabolic pathway. This then shows, instead of showing you the mechanism, this shows you the actual product name or program name. Again, start at the bottom right. We have 3 ways to replace CBS: through pegtibatinase, through gene therapy, or chaperone therapy to stabilize the defective enzyme. Then lower left, homocysteinase, the Aglia program, replacing CBS. Then the upper left, the 2 GI therapies, and then the 2 metabolic therapies, Formate, acts right where betaine does in that part of the pathway, and the thiol-based reductants would directly affect homocysteine and enable more to come down that lower pathway. Now, I'm not going to go through this map, but years ago when we first came up with the research map and strategy, we wanted to have a way to depict it on one slide, so that if someone just wanted a summary of what are the therapies, where are they in development, when could they get to market, they could look at this. So I'm pleased to say that we're now showing things getting to market within the 5 or 6 year time frame. I'm also pleased to say the map is now 2 pages. So we've made a lot of progress, and we're really excited about that. this is on our website, not this current version. I just updated it for the meeting. We will have this on the website within the next week or two. We're going to let Danae take a few days off. So I've alluded to our Global Grants Program. I'll just quickly explain how this works. So back in 2016, we had a project to develop a research map. What are all the studies that have been done for HCU? Which ones showed a positive result? Categorize them. We then convened a global scientific advisory board. They then identified the key priorities. We established a global grants process. We did this in partnership with HCU Network Australia, since Tara Morrison was so engaged and involved in helping us at a global level. We have now had 3 calls for grants, and the same 2 priorities were determined by the SAB for all 3 calls. First and foremost, new treatment approaches, and second, an improved approach to newborn screening. The results of this grants program, we've awarded 3 grants now for potential new treatment approaches for CBS deficient HCU. I won't read you those. I've talked about all 3 of them as I went through the programs. And actually, I should say that we're just today announcing, we have not yet publicly announced the Thomas McCorvey grant. Danae will be issuing that press release within the next couple of days. We did not fund any proposals for newborn screening because none of them were deemed to be a first-tier screen. And so we already have a viable approach with a 2-tier screen. We just have to keep working on getting states to adopt that. We're up to 6 or 7. Danae's been working very hard on that with support from some of our partners. But we're going to continue to work at that. And the good news is the CDC has announced that they've come up with an approach. We've been working with them as well, and they are now just validating this approach. And it could mean that you can measure homocysteine right at that first newborn screen, which is going to have a significant increase in the number and percentage of babies who are detected. We're also thrilled that we've significantly grown the number of proposals coming in from 2 to 5 to 11. And so that's great. There's a lot more researchers now interested in this field and submitting more proposals. And the other important thing on this slide, we agreed early on, as HCU Network America originally was formed to focus on CBS deficient HCU. That was a focus for our grants program early on. But over time, as we learned about the unmet needs with some of the cobalamin disorders and agreed that we would expand our focus, cobalamin and severe MTHFR, we did slightly generalize our 3rd call, and we got a really good proposal for a cobalamin C program, which also is looking at chaperone therapy, like I explained, combining with that defective enzyme. The problem was, when we fundraise, if we get funding coming in for families with CBS deficiency, then they would expect that funding to go towards research for CBS deficiency. And we've just really started to engage and have fundraising from some of the cobalamin severe MTHFR. So we have had some funding to allocate, but not enough to give a $40,000 grant. And it was Brittany's suggestion. I don't know if it's Brittany in the room. She might be in another meeting right now. But Brittany Park, who leads our cobalamin steering committee, said, well, gee, why don't we talk, the Organic Acidemia Foundation would be willing to co-fund this. And then through Tara, we met the CBLC ONLIS group in Italy, where they have a high number of cobalamin C families, and they were willing to co-fund it. So it was amazing that we got 3 organizations to all agree on who would pay what. CBLC ONLIS contributed the most to agree on the contract and the press release, and this work is ongoing right now. And so, as Brittany just presented to the cobalamin MTHFR families, we are going to set up a separate grants process because this itself is huge to manage. And I say that as the one who manages it. And also, you want experts in cobalamin and severe MTHFR. And while some of our panel has some expertise, there are others who deal exclusively with that. So Brittany will be carrying that forward and helping to do more fundraising. And so what else do we need to do, and how do we need your help? Well, first, we need to continue to increase fundraising so we can fund more and more research, and maybe that 2-page map could extend to 3 pages. But we also need to extend that fundraising to include remethylation disorders. We need to continue to liaise with industry and academia. We're able and willing to help any researcher or organization that wants to bring forward therapies for these disorders, and Danae spends a lot of time doing that. And we encourage all of you as members of the HCU patient community to support this research. It doesn't work unless there are HCU patients who are enrolled. Natural history studies, Phase 1, Phase 2 studies, soon to be Phase 3 studies, other supporting programs like RareRx, having the information that researchers need to even plan their hypotheses or research programs. Coriell Biobank, having blood samples that can help provide support to researchers looking to work in this area. None of this will work without all of your support, so visit that breakout room over there if you haven't already met with the phlebotomist. And so I'd like to end by thanking all those who made this possible. First, those I would consider co-sponsors. Tara has been a great partner, and she co-funded rounds 1 and 2. She's been funding other things like the global patient meeting and global expert meeting. So wasn't part of round 3, but we know she'll be back. But then we had new partners emerge for our Cobalamin C grant, which was great. The leaders of our scientific advisory board who put a lot of time into this. Victor Kosick was the chair for 4 years. Warren Kruger was the co-chair. Warren has now taken over as chair, and Andrew Morris from the UK has come on as the new co-chair. These researchers spend a lot of time to help us with this. And then members of the scientific advisory board, including Kim Chapman, who I know just walked back in the room back there. So thank you, Kim, for all the time you devote to this. Bridget Wilkin recently retired, and actually we've just got agreement from Kostov Bhattacharya in Australia, who's going to replace Bridget in that role. And we unfortunately lost John Krause, who was a very dedicated member of this group. And then everyone involved in the research, the universities, the companies, the clinicians, and the patients who support these trials. And lastly, as my introduction by Kristen explained, I lost these 2 sisters back in, I think it was 1972, within 6 months. Judy died of a stroke at age 14, and Susie of pulmonary embolism at age 9. And I always said, I always said it was so unfair there were no treatments available. My father owned a pharmacy. He had medications for hundreds, if not thousands, of other diseases. And as a kid, I said, why aren't there any treatments available for my sisters? And I vowed someday I'd do something about it, and I'm glad to be part of this whole effort to help bring about those new therapies. I also apologize to Judy. If she were looking at this picture, she'd say, why do you show the worst picture of me? And I'm showing it because that's what HCU patients look like before we really knew how to deal with their issues. And she had the Marfanoid type, long, thin limbs, very, very bad ophthalmic issues, etc. Susie was a little bit healthier. And as someone said earlier, the phenotypes of how the same exact disease, they had the exact same defect, how it appears in its siblings can be different. But I thank them for the inspiration.